Hello everyone, welcome to Aesthetic Food Tech. So, this is our second video about protein determination, and in this video, we are going to discuss about quantitative determination of protein. Quantitative determination of protein is protein determination based on amount or quantity. It is also called as indirect determination of protein. This is because in this protein content or protein is determined based on nitrogen content. The quantitative method for determination of protein is Geldahl method. Now, let us discuss about Geldahl method and let us begin with an introduction. Geldahl method was developed in 1883 by a scientist called John Geldahl, and in this, protein content is determined quantitatively based on nitrogen concentration. The principle of Geldahl method is that a food is digested with a strong base, and as a result, nitrogen is produced or released. This nitrogen can be determined based on a suitable titration technique, and protein content can be determined from this nitrogen concentration. This method is considered as standard method for the determination of protein concentration, and also in this, we are not determining the concentration of protein directly, but a conversion factor is used to convert the nitrogen concentration into protein concentration. Usually, 6.25 is the value of this conversion factor, which is expressed as F. But it differs from each protein depending on the composition of amino acids. Next topic, steps in Geldahl method. Geldahl method is mainly divided into 3 steps, and they are first one digestion, second one distillation, and third one titration. So let us discuss about each step in detail. First step is digestion. In this step, the sample to be analyzed is weighed into a digestion flask, and it is digested by heating in the presence of sulfuric acid and anhydrous sodium sulfate. Catalysts such as copper, titanium, mercury, selenium is used. Digestion converts nitrogen into ammonia and other organic matter into carbon dioxide and water. Ammonia gas is not produced as a result of this digestion process. It is because ammonia is in the form of ammonium ion, which binds with sulfate ion and remain as ammonium sulfate. Distillation. After digestion, the digestion flask is connected to a receiving flask using a tube. And we know that in digestion we used sulfuric acid. So definitely the solution will be acidic. That is, the solution remained in the digestion flask will be acidic. And for converting this acidic solution to basic or alkaline solution, sodium hydroxide or NaOH is used. And as a result of using NaOH, the ammonium sulfate which was present in digestion flask will get converted to ammonia gas. The reaction is clearly mentioned below. You can see and learn it. The ammonium gas formed get liberates and enter into receiving flask. Receiving flask contain boric acid, and this receiving flask convert ammonium gas into ammonium ions and boric acid into borate ions, and as a result they remain as ammonium borate. The reaction is clearly mentioned. You can see it. Last step is titration. Nitrogen content is determined by titrating ammonium borate with standard sulfuric acid or hydrochloric acid. Suitable indicator is used to determine endpoint in the titration. Let us look for the equation that is used to calculate the nitrogen percentage. Percentage of nitrogen equal to x moles divided by 100 centimeter cube into Vs minus Vb into 14 gram into 100. X is the mass of sample, 14 is the mass of nitrogen, Vs is the titrated value of sample, and Vb is the titrated value of blank. Using this nitrogen percentage of protein is calculated by using the equation percentage of protein equal to F into percentage of nitrogen. F is the conversion factor that we discussed while discussing about the principle of Geldahl method. Next topic, advantages of Geldahl method. First one, it is widely used internationally as standard method. Second one, it has high precision and good reproductivity. Please forgive for my spelling mistake. I had wrote highly instead of high in the note. Disadvantages of gel dal method. First one, it is time consuming. Second one, different conversion factor is used for different protein. Third one, hazard causing due to the use of concentrated H2SO4 at high temperature. That is, we use concentrated H2SO4 at high temperature during digestion process. Please keep it in mind.